Fine, let's start. Number 1. The Sima de los Huesos, Denisovan mitochondrial genome. We begin underground, in the limestone galleries of the Sierra de Atapuerca in northern Spain. Deep inside this cave system lies a vertical shaft that researchers call the Sima de los Huesos, the pit of bones. The name is precise. At the bottom of that shaft, excavators have recovered the remains of at least 28 Middle Pleistocene humans, making it one of the richest early human fossil collections anywhere on Earth. These are not scattered fragments. They include skulls, jaws, teeth, and postcranial bones in remarkable abundance. And they are old, dating places the Sima de los Huesos hominins at roughly 430,000 years before present, far older than most successful ancient DNA recoveries. For decades, the bones spoke for themselves, and what they said seemed clear. The anatomy looked Neanderthal-like, with the heavy brow ridges and facial features that researchers associate with the Neanderthal lineage. Then the genetics arrived, and the bones and the molecules began to disagree. Recovering DNA from material, this ancient is extraordinarily difficult, so scientists targeted mitochondrial DNA first. The reason is a matter of arithmetic. A cell carries only 2 copies of its nuclear genome, but it can carry hundreds or thousands of mitochondria, each with its own small loop of DNA. More copies mean better odds of recovery from degraded tissue. The team managed to reconstruct a nearly complete mitochondrial genome from a Sima individual. That completeness mattered. A tiny fragment can be dismissed as noise or contamination. A near-complete sequence is much harder to wave away. Here is the expected result. Neanderthal-like anatomy should have produced Neanderthal-like mitochondrial DNA. Here is the actual result. The Sima mitochondrial genome clustered closer to Denisovans, a population then known mainly from a few remains in a Siberian cave thousands of kilometers to the east. A Spanish skeleton that looked Neanderthal carried a maternal lineage pointing toward Siberia. To understand the puzzle, you have to understand a limit of the evidence. Mitochondrial DNA is inherited only from the mother. It traces a single maternal thread, not the whole tapestry of an organism's ancestry. So the Sima result was real, but it described one line, not the entire family. Later work resolved part of the contradiction. When researchers, including teams associated with Svante Pääbo and Matthias Meyer, recovered nuclear DNA from the Sima population, that nuclear signal placed the group firmly on the Neanderthal lineage. This gives us the central technical concept of the section, mitochondrial nuclear discordance. Maternal ancestry and whole genome ancestry can tell different stories about the same body. One skeleton can preserve 2 histories at once. How does that happen? Researchers proposed several mechanisms: ancient population structure, in which deeply divided groups exchanged genes; incomplete lineage sorting, in which old genetic variants survive unevenly across descendant populations; replacement of an earlier mitochondrial lineage by a later one; or gene flow that introduced new maternal DNA after the populations had begun to separate. What was lost here was a maternal lineage. The mitochondrial branch visible at Sima de los Huesos did not survive into the later classic Neanderthals, whose mitochondrial DNA looks different. Fossil anatomy could never have revealed that disappearance, because bone shape does not record which maternal line a population carried. The Denisovan side of human history was nearly invisible until DNA exposed it. In modern terms, this is why ancient DNA now routinely rewrites family trees built from fossils alone. The Sima result also pushed the estimated split between the Neanderthal and Denisovan lineages deeper into the past, beyond 430,000 years ago. And it gave researchers a working image of human evolution as reticulated, meaning a network of branches that separate and rejoin through interbreeding, rather than a tidy tree with clean, permanent forks. The controversy was never whether the sequence existed. The controversy was what population history could produce it. The bones said Neanderthal. The maternal genome pointed east toward a ghost lineage. So if a ghost lineage can hide inside Neanderthal bones, what happens when there are no bones at all? Number 2. The ghost archaic admixture in West African genomes. This time we do not begin in an excavation trench. We begin in modern West Africa, among living people. The genomes in question come from present-day populations. The Yoruba, the Isan, the Mende, and Gambian groups, all sampled as part of large international genetics projects. To read this case correctly, set aside one common assumption. Africa is often described as the simple origin point of humanity, a single starting line. The genetics suggests something more complex. Africa holds the deepest reservoir of human genetic diversity on the planet, and that diversity hints at a long, branching internal history. The key term is ghost population. A ghost population is a human lineage, inferred from DNA, but not yet confirmed by any fossil. It is detected, not dug up. Researchers find it by studying patterns in living genomes. The site frequency spectrum, which describes how common different genetic variants are. Unusual haplotypes, meaning blocks of DNA that look strangely divergent. And segments of the genome that diverge far more deeply than expected. When statistical models work through these patterns, they can point to a contribution from a population that no longer exists. For West Africa, the estimated archaic contribution falls in a broad range, roughly 2% to 19%, depending on the population and the model used. That spread is important. The signal is not uniform across all West Africans, and it should not be flattened into a single dramatic number. The estimated timing of this admixture is often discussed in terms of tens of thousands of years ago, with figures near 50,000 years frequently cited. The central study here is the 2020 work by Arundur Vasala and Sriram Sankararaman, who built a statistical inference model to test for this hidden contribution. Their analysis suggested that the inferred archaic lineage may have split away before Neanderthals and modern humans had fully diverged from one another. Compare this with the better-known case of Eurasian archaic admixture. In Eurasia, scientists have both fossils and sequenced genomes for Neanderthals and Denisovans. In West Africa, they have a statistical signal and no confirmed skeleton to attach it to. The reason is environmental. Heat and humidity are hostile to DNA preservation. Across much of Africa, the molecule simply does not survive long enough to be recovered. So how is this knowledge lost? The archaic population may have left no recoverable skeletons and no distinct cultural signature that excavators could trace. There is also a serious analytical caution. Deep population structure within Homo sapiens itself, meaning groups that were long separated, but never a different kind of human, can produce statistical patterns that resemble admixture from a separate archaic lineage. Telling those two scenarios apart is genuinely hard, and a fossil-free lineage is easy to misunderstand or to sensationalize. The careful interpretation is restrained. Africa's human past was likely multilineage and regionally complex. The category modern human in Africa may describe a network of interacting populations rather than a single, clean ancestral group. Researchers hope that sediment DNA, the study of ancient proteins known as proteomics, and improved preservation techniques will eventually give this lineage a physical form. For now, the hidden lineage survives only as mathematical residue in the genomes of living people. The most controversial fossil in West Africa may be the one no one has found, but not all archaic DNA stays hidden. Sometimes it does something visible and useful. Number 3. The Denisovan EPAS1 high-altitude gene in Tibetan populations. Consider the Tibetan Plateau as a survival test. Much of it sits above 4,000 meters in elevation. At that height, the air holds far less oxygen, and permanent human life requires real biological adaptation to chronic oxygen shortage, a condition called hypoxia. This is not a minor inconvenience. Hypoxia affects pregnancy outcomes, circulation, physical work capacity, and basic survival. A population that lives on the plateau generation after generation is a population that has solved this problem. The gene at the center of the story is EPAS1, which sits in the body's hypoxia response pathway and helps regulate how the body handles oxygen. To see why the Tibetan version is unusual, look at what most lowlanders do at altitude. A typical lowland response to thin air is to produce more hemoglobin, the oxygen-carrying component of blood. More hemoglobin sounds helpful, but in excess, it thickens the blood and carries health costs. Tibetans show a different pattern. They manage oxygen efficiently, without the extreme hemoglobin elevation seen in many other groups. When researchers examined the Tibetan version of EPAS1, the haplotype, meaning the specific linked block of DNA, looked strikingly divergent from ordinary modern human variation. It did not fit the normal range. Then came the shock. The Tibetan EPAS1 haplotype matched Denisovan-like DNA better than it matched the available modern human sequences. Genome comparison, including work led by Emilia Huerta Sanchez and Rasmus Nielsen, revealed that this was a case of adaptive introgression. Introgression means DNA that entered one population from another lineage through interbreeding. Adaptive means that the inherited DNA proved useful, so natural selection favored it. The haplotype is rare in Han Chinese populations, but common in Tibetans. The most likely sequence of events is a learning curve in 2 stages. First, the variant entered the modern human population through ancient interbreeding with Denisovans. Then, after people settled the high plateau, the survival pressure of low oxygen amplified that Denisovan-like variant, driving it to high frequency in Tibetans specifically. What was lost is striking when set against what survived. Denisovans left very few physical remains, a handful of bones and teeth, yet their biological influence is substantial. The gap between the tiny fossil record and the enormous functional consequence is one of the strangest features of this case. It also complicates the idea of firm species boundaries. A genuinely useful gene crossed from an archaic human group into Homo sapiens and stayed. In modern terms, EPAS1 now feeds directly into research on altitude physiology, pregnancy at elevation, and oxygen stress in medicine, and it overturns an old assumption. Archaic DNA was not always harmful evolutionary baggage to be purged. Sometimes it was a toolkit. Denisovan ancestry, in varying forms, appears in multiple populations across Asia and Oceania. One vanished population helped another survive one of the harshest habitats on the planet. Denisovans barely left bones, but they left Tibetans a genetic oxygen system. Yet for every finding that strengthens with scrutiny, there is one that collapses under it. Number 4. The Lake Mungo man mitochondrial DNA controversy. This is a cautionary case. It begins in the Willandra Lakes region of New South Wales in Australia, a dry landscape of ancient lake beds. From there came a set of remains designated Lake Mungo 3, also recorded as WLH3, and known publicly as Mungo Man. The remains are roughly 40,000 to 42,000 years old, an early and significant record of human presence on the Australian continent. In 2001, a research team published a claim that they had extracted mitochondrial DNA from these ancient Australian remains. The interpretation was dramatic. The recovered sequence appeared to fall deeply outside the mitochondrial diversity of all living modern humans. If true, that would be a serious problem for simple versions of the recent Out of Africa model, in which all living people descend from a single, relatively recent African population. A maternal lineage older and more divergent than anything in living humans would imply a far more tangled origin. To judge the claim, you need to understand the method of the time. Early ancient DNA work relied heavily on the polymerase chain reaction, or PCR, a technique that copies tiny fragments of DNA over and over until there is enough material to read. PCR is powerful, but it has a dangerous blind spot. It amplifies whatever DNA it is given. It cannot tell ancient target DNA from modern contaminating DNA. If a few modern molecules slip into the sample, PCR will faithfully multiply them alongside, or instead of, the genuine ancient material. The correction came in 2016. A reanalysis associated with researchers including Tim Lambert and the laboratory of Alan Cooper applied newer sequencing and DNA capture methods to the Lake Mungo material. The result was deflating. The reanalysis found no identifiable Aboriginal Australian DNA in the WLH3 sample. Instead, it detected several modern European contaminant mitochondrial sequences. The extraordinary 2001 finding had not revealed an ancient lineage. It had captured modern human contamination. This is why authentication standards tightened. Today, a credible ancient DNA result is expected to show characteristic chemical damage patterns, short fragment lengths typical of old DNA, independent replication, and formal estimates of contamination. The Lake Mungo episode helped make those checks mandatory. Why was the genuine signal lost in the first place? Australia is warm, and warmth drives DNA to fragment and degrade. On top of that, every stage of an artifact's life, the excavation, the handling, the museum curation, and the laboratory work itself, offers a chance for modern human DNA to enter the sample. The remains were also culturally and politically sensitive, which made the original claim even more explosive when it was published. The lasting value of Lake Mungo is procedural. It shows how fragile evidence, taken at face value, can briefly redirect the story of human origins. A sequence once threatened to rewrite human prehistory. Later, it became a lesson in contamination. Contamination distorts the past in one way. The next case shows how probability can be distorted into something it never claimed to be. Number 5. The Cheddar Man pigmentation data. Here genetic probability collided with public identity. The remains belong to Cheddar Man, found in Gough's Cave in the Cheddar Gorge of Somerset, England. He is roughly 10,000 years old, a man of Mesolithic Britain. In terms of ancestry, he belongs to a group geneticists called Western hunter-gatherers, the population present in Europe before the later arrival of Neolithic farmers, and, later still, migrants associated with the Eurasian steppe. In 2018, researchers at the Natural History Museum in London, working with geneticists including Ian Barnes and Selena Brace, produced a reconstruction of Cheddar Man based on ancient DNA. The widely reported conclusion was that he likely had dark to dark brown skin, dark hair, and blue or blue-green eyes. To interpret this responsibly, hold one fact in mind. DNA does not deliver a photograph. It delivers probabilities. Forensic pigmentation tools work by comparing DNA markers from a sample against large modern datasets that link genotypes to observed pigmentation. From that comparison, they generate likelihoods, not certainties. The biology underneath is polygenic. Skin color is not controlled by a single switch. It depends on many genes acting together, which means a prediction is a weighted estimate across numerous markers. The natural history. Museum's analysis placed Cheddar Man outside the lightest pigmentation categories, and that conclusion was consistent with other findings for Western hunter-gatherers across Europe. It is also consistent with a learning curve in European pigmentation. Alleles associated with lighter skin became more common in northwestern Europe later, carried in by subsequent migrations of farmers and steppe populations. Cheddar Man sits before much of that change. Uncertainty remained, though, from missing genetic markers, low sequencing coverage in places, and the inherent limits of the predictive models. What was lost is everything soft. No skin, no hair, no surviving soft tissue remained to verify the prediction directly. The reconstruction was an inference rendered as a face. And there is the difficulty. A physical model, a sculpted head with a definite color, can make a probabilistic finding look like a settled fact. The public debate in Britain became political. because the image touched questions of national identity and the imagined appearance of the first Britons. In modern terms, this case shows how ancient DNA now feeds directly into arguments about ancestry, migration, and belonging. The defensible scientific claim is narrow. It is a statement about the most likely pigmentation category, not a guarantee of an exact shade. The science was probabilistic. The public reaction treated it as symbolic. Cheddar Man became controversial because DNA challenged the face people expected their history to have. A single skeleton can be stretched in this way. So can a single cemetery. Number 6. The Schuenemann et al. Ancient Egyptian Mummy Genome. This is a story of a real technical breakthrough, forced to carry a much larger argument than its data could support. The setting is Abu Sir el-Malek, an archaeological site in Middle Egypt. The samples span a long stretch of time, roughly 1,300 years, reaching from the New Kingdom through to the Roman period. Ancient Egyptian ancestry is an unusually charged subject because it is tied to African, Mediterranean, Near Eastern, Arab, and modern national identities, all at once. The study in question, published in 2017 and led by Verena Schuenemann, together with Johannes Krause and colleagues, was a major achievement in mummy DNA recovery. The dataset deserves precise description. The team obtained 90 mitochondrial genomes and genome-wide nuclear data from only 3 individuals. That gap between 90 and 3 matters, and we will return to it. Mummies were long considered nearly impossible targets for ancient DNA. The reasons were heat, the chemicals used in embalming, regional humidity, and centuries of handling. The team worked around this in part by sampling dense bone and tooth material, where DNA survives better and surface contamination is less of a threat. 2 kinds of data appear here, and they are not equivalent. Mitochondrial DNA traces only the maternal line. Genome-wide nuclear data samples ancestry across the whole genome. 90 maternal genomes is a substantial dataset. 3 genome-wide samples is a very thin basis for any claim about an entire civilization. The main finding was that the Abu Sir el-Malek samples showed a strong genetic affinity to ancient Near Eastern populations. The study also reported that modern Egyptians, in their models, carried more sub-Saharan African-related ancestry than the ancient samples did, with the increase estimated at roughly 8%. What was lost, or rather never sampled, is the rest of Egypt. Abu Sir el-Malek is one site in the middle of the country. Southern Egypt, Nubia, the desert oases, and the frontier zones could carry different genetic profiles, and they remain undersampled. Mummified remains can also reflect the biases of a particular cemetery, perhaps an elite group or a specific local community, rather than a national average. In modern terms, this case is a lesson in why sample geography matters. Future genomes from other regions and other periods may complicate the picture considerably, and one phrase needs careful handling. Closer to Near Easterners is a statement about statistical affinity. It does not mean not Egyptian, and it does not mean European. The study answered the history of one cemetery far better than it answered the history of a civilization. Ancient Egypt's DNA became controversial because a single narrow dataset was asked to settle a 1,000-year-old identity argument. Identity also drives the next case, where a real genetic puzzle was seized on by people looking for a very different answer. Number 7. Haplogroup X in Native American populations. Native American maternal ancestry is traditionally described through 5 mitochondrial haplogroups, labeled A, B, C, D, and X. A haplogroup is a branch of the maternal family tree, traced through mitochondrial DNA. 4 of those branches are relatively straightforward. The 5th, haplogroup X, became the center of a long argument. Haplogroup X appears in only a minority of Native American populations, often discussed as being present at a level of a few percent. The branch that matters is X2A, the distinctive Native American sub-lineage of haplogroup X. The broader haplogroup X is found across Eurasia, including in Europe and the Near East. That wide distribution is what attracted attention. But here is the key technical point. Native American X2A is not a simple match to modern European or Middle Eastern X lineages. It is its own distinct branch. The real puzzle is not a transatlantic mystery. It is the absence of obvious intermediate lineages. There is a gap in Siberia, or Beringia, the land bridge region between Asia and the Americas, where the connecting lineages either went extinct or have not been found. The explanation that geneticists favor is founder effects and bottlenecks. When a small group moves into a new region, it carries only a slice of the original genetic variation. A rare lineage can survive in that new region while vanishing from the source population. Researchers analyzed X2A using phylogeography, the study of how lineages are distributed in space, along with coalescence dating, which estimates when lineages shared a common ancestor, and direct comparison with ancient DNA. The decisive evidence is that pre-contact remains. Individuals who lived in the Americas before any European arrival carry X2A-related ancestry. That rules out the idea that X2A was introduced by colonial-era Europeans. Against this stood the Solutrean hypothesis, which argued that Ice Age Europeans crossed the Atlantic and influenced the early peopling of the Americas. The hypothesis leaned partly on haplogroup X. Geneticists generally reject X2A as evidence for a European founding population. How was the connecting knowledge lost? Beringian populations may have vanished, migrated away, or been absorbed into later groups. Subsequent genetic turnover across Siberia could have erased the source populations that once carried X2A's relatives. The crucial logical point is this: the absence of modern Siberian X2A relatives is not evidence of a European origin. It is a gap, and a gap is not a doorway. Ancient genomes, including the Anzac 1 individual we will reach shortly, allowed researchers to test early ancestry directly rather than guess from haplogroup distributions. This case shows how a genuine scientific anomaly can be exploited by fringe narratives. X2A remains genuinely interesting, but it is not evidence that Europeans founded the Americas. The mystery is not Europe and America. It is a lost Beringian maternal branch. To see how a single finding can topple an entire old framework, we turn to the discovery that changed everything. Number 8. The 2010 Neanderthal interbreeding discovery. For most of the 20th century, the dominant picture placed Neanderthals on a separate side branch of the human tree, a related group that was replaced by modern humans and left no descendants. Around that picture ran a long debate between competing models. The replacement model held that modern humans simply replaced archaic groups with little or no interbreeding. The multi-regional model held that modern humans evolved across several regions at once, with gene flow between them. The assimilation model sat in between, allowing for some absorption of archaic populations. Neanderthals themselves were Eurasian humans who lived for hundreds of thousands of years before disappearing from the record. Sequencing Neanderthal DNA was a formidable technical challenge. The DNA fragments recovered from Neanderthal bones were short, chemically damaged, and heavily mixed with the DNA of microbes that had colonized the remains after death. A particular danger was contamination by modern human DNA, since Neanderthal DNA is so similar to ours that telling them apart requires great care. The team developed methods to filter the genuine Neanderthal signal out of that noise. Then they compared the Neanderthal genome with the genomes of living people. The pattern was clear and unexpected. Neanderthal genomes shared more genetic variants with non-Africans than with sub-Saharan Africans. The most economical explanation was interbreeding. The original estimate placed Neanderthal ancestry at roughly 1% to 4% of the genome in non-African humans. This was the 2010 draft Neanderthal genome, produced by the team led by Svante Pääbo at the Max Planck Institute for Evolutionary Anthropology. The admixture most likely happened before the major Eurasian populations split from one another, and the refined timing is often placed around 50,000 to 45,000 years ago. Peibo would later receive the Nobel Prize in Physiology or Medicine, awarded in 2022 for his work founding the field of paleogenomics and sequencing extinct hominin genomes. What was lost was Neanderthal identity as a distinct group. Neanderthals did not survive as a separate population. They survived as fragments distributed through the genomes of billions of living people. Morphology alone could never have proven interbreeding at this scale, because a bone cannot show you which segments of a genome were inherited from whom. Some experts initially resisted the result, precisely because it overturned decades of careful interpretation. The modern relevance is broad. Neanderthal-derived DNA influences immune function, skin biology, metabolism, and the risk of certain diseases in living people. The picture is not uniform across the genome. Some regions are noticeably depleted of Neanderthal DNA, a sign that natural selection removed segments that were harmful in a modern human background. Other regions retained Neanderthal DNA, possibly because those segments offered an advantage. Neanderthals became ancestors, not merely evolutionary cousins. One genome made extinction less absolute than anyone had expected, and once direct genomes were available, they could be used to test old archaeological claims head-on. Number 9. The Anzic One child and the destruction of the Solutrean hypothesis. The Anzic One individual is the infant remains of a child found in western Montana in the United States. The burial is roughly 12,600 to 12,700 years before present. The context is what makes it famous. The child was buried with artifacts associated with the Clovis culture, one of the most discussed early archaeological traditions of North America, known for its distinctive fluted stone projectile points. Researchers, in a team led by Eski Willerslev, sequenced the genome of this Clovis-associated child. The power of the study lay in its directness. Instead of inferring ancestry from the style of stone tools, the team could test ancestry from the genome itself. The claim under examination was the Solutrean hypothesis. That hypothesis, advanced most prominently by archaeologists Dennis Stanford and Bruce Bradley, proposed that Ice Age people from the Solutrean culture of southwestern Europe crossed the Atlantic and contributed to, or even founded, the Clovis tradition. The hypothesis rested on perceived similarities between Solutrean and Clovis stone tools, and it drew support from the Haplogroup X argument we examined earlier. A genome could test something a stone tool could not. Whether Clovis ancestry carried any special European connection, the result was decisive. The Anzic One child showed strong ancestry ties to indigenous peoples of the Americas, with particular affinity to native populations of Central and South America. There was no special European ancestry signal of the kind the Solutrean hypothesis required. Instead, the genome connected Anzic One to Northeast Asian and Beringian-related origins, consistent with entry into the Americas from Asia. The negative result was as important as the positive one. How does an archaeological culture vanish in a way that leaves room for such debate? Clovis culture itself disappeared, transformed, or was replaced by later regional traditions, so the people who made those tools did not continue as an identifiable group with an unbroken label. This points to a deeper lesson about evidence. Tools can spread, change, and even converge on similar designs without any movement of people, and without genetic replacement. Cultural similarity is simply weaker evidence than genome-wide ancestry. The Anzic One study also raised serious ethical questions, and the researchers engaged in consultation with descendant Native American communities, addressing repatriation and the proper treatment of ancestral remains. The genome reframed the Clovis people as relatives of later indigenous populations, rather than as a separate founding stock. Some Solutrean proponents continued to argue their case even after the genetic evidence. The genome did not answer every question about how the Americas were peopled, but it crippled the European Clovis claim. A child's DNA ended a theory that stone tools had kept alive. If genomes can close an old crossing, they can also reveal one that history forgot. Number 10. The Pre-Columbian Polynesian-Native American Admixture signal. The setting is the open Pacific, the vast triangle of eastern Polynesia, with Rapa Nui, also known as Easter Island, marking its far eastern edge. For a long time, scattered clues hinted at contact between Polynesia and the Americas. The sweet potato, a plant native to the Americas, was cultivated across Polynesia long before European voyages. There were voyaging traditions, sophisticated canoe navigation, and debated linguistic parallels. The central question was direct. Did Polynesians and Native Americans meet before Europeans arrived in either region? Genetics offered a way to test it. Researchers carried out genome-wide admixture analysis on modern Polynesian populations, looking for segments of DNA inherited from a different ancestral source. The signal appeared. Native American-related ancestry shows up in several Polynesian populations. Crucially, the genetic dating placed this admixture at approximately 1200 CE in parts of eastern Polynesia. That date sits well before Columbus and well before sustained European contact. Some models pointed to an affinity with groups from northern South America. The major modern study, published in 2020 and led by Alexander Ioannidis with Andres Moreno Estrada and colleagues, analyzed genome-wide data from hundreds of Polynesian individuals. A reasonable objection to any modern DNA result is contamination from recent, post-European mixing. So, a stronger test followed. In 2024, researchers published ancient genomes from Rapa Nui itself. They sequenced 15 ancient Rapa Nui individuals, dated roughly to between 1670 and 1950 CE. The ancient data showed about 10% Native American admixture, with modeling that placed the contact event around 1250 to 1430 CE. Ancient genomes, less exposed to recent mixing, supported the pre-Columbian timing. Why was this contact so hard to see for so long? Voyages across the open Pacific left little durable archaeological evidence. Canoes were made of perishable material. Coastlines erode. Contact groups may have been small, leaving a faint footprint. Before DNA, the case rested on oral memory and the distribution of plants like the sweet potato, both of which were debated. The genetics strengthened the case considerably. One question remains genuinely open: the direction of contact. Did Polynesians sail east to the Americas? Did Native Americans sail west into the Pacific? Or did contact go both ways? Polynesian navigation skill makes eastward voyaging plausible, but genetic data alone struggles to fix direction. Future ancient genomes from South American coastal sites and from earlier Rapa Nui burials could refine the answer. The Pacific was not a wall, it was a navigated space. The DNA proves that contact happened, while the ocean still hides who made the crossing first. That completes the 10 findings, but the controversies share a common engine. And to understand them fully, you need to understand the tool that produced them. So let us look at ancient DNA itself. Ancient DNA transformed archaeology. For most of the discipline's history, the evidence was objects, bones, stone tools, pottery, and the patterns in which they were buried. Researchers read those objects to infer who lived where. Ancient DNA shifted the focus to genomes, haplotypes, and admixture dates, allowing direct testing of ancestry rather than inference from artifacts. The oldest and most controversial claims tend to come from places where preservation is poor, because that is where the signal is weakest and the room for error is largest. The core problem is degradation. After death, DNA breaks down. The strands fragment into ever shorter pieces. The molecule suffers chemical damage. Microbes invade the tissue and add their own DNA to the mix. environmental conditions accelerate all of this. One specific and useful damage pattern is the deamination of cytosine, a chemical change to one of the DNA bases that occurs predictably at the ends of ancient fragments. That pattern has become a signature. When researchers see it, they have reason to believe a sequence is genuinely old rather than a modern contaminant. Authentic ancient DNA is also characteristically short and broken, so fragment length itself is a clue. To find the best preserved material, researchers target teeth, the dense petrous portion of the temporal bone near the inner ear, and other dense skeletal tissue, where DNA survives longest. And they always distinguish between mitochondrial DNA, which traces the maternal line, and nuclear DNA, which records genome-wide ancestry. Different sites in this script illustrate different preservation challenges. Sima de los Huesos tested the limits of age at over 400,000 years. The Neanderthal genome tested the problem of separating archaic DNA from modern contamination. Lake Mungo showed what warm climate degradation does. The Egyptian mummies showed the combined obstacles of heat, embalming chemistry, and handling. Once DNA is recovered, the analysis brings its own demands. Sequencing depth and coverage, formal contamination estimates, and careful choice of which modern and ancient populations to use as references. The statistical toolkit includes principal component analysis, which arranges samples by genetic similarity, F statistics, and D statistics, which test specific models of population relationship and gene flow, haplotype sharing analysis, and Bayesian methods for dating events. A central caution follows from all this. The same genetic data, run through different models with different reference populations, can produce different answers. The model is part of the result. This also explains why some regions are nearly silent. Warm regions, acidic soils, and repeated handling all destroy DNA. Tropical Africa, Australia, and Egypt are far harder than the cold, dry caves of Eurasia. And the absence of recovered DNA is not the absence of people or history. It is the absence of a surviving signal. Newer methods are extending the reach of the field. Sediment DNA, which recovers genetic material directly from cave floors without any skeleton. Proteomics, the study of more durable ancient proteins. Improved extraction chemistry. And single-stranded library preparation, which captures shorter and more damaged fragments than older methods could. These advances may yet give a voice to currently silent regions. Ancient DNA is not magic. It is damaged evidence rescued by probability. The method can reveal lost humans, but only when the signal survives the noise. And when the signal is faint, the risk is not silence. The risk is overconfidence. That risk deserves its own examination, because ancient DNA controversies often begin at a specific point, where technical uncertainty meets public certainty. Lake Mungo is the central cautionary case for contamination. Cheddar Man is the central cautionary case for probabilistic phenotype prediction. Both show how a careful, hedged scientific statement can harden into a confident public claim. Contamination, in technical terms, is modern DNA introduced into a sample during excavation, storage, handling, or laboratory work. Human DNA contamination is the most dangerous kind, for a simple reason. The target is also human. A microbial contaminant is easy to spot because it looks nothing like the sample. A modern human contaminant in an ancient human sample blends in. Laboratories develop defenses, dedicated clean rooms, protective clothing, blank control samples that should contain no DNA, independent replication in separate labs, and authentication based on those cytosine damage patterns. Prediction carries a different kind of uncertainty. Pigmentation estimates, ancestry percentages, and admixture dates are all outputs of models. They are not direct readings, and they depend on reference bias. The result changes depending on which modern and ancient populations are chosen for comparison. The contrasts are instructive. The 2001 Lake Mungo claim made with PCR-era methods, was overturned by the 2016 reanalysis with better tools. The Schuenemann study's 3 genome-wide samples from one cemetery were sometimes stretched, in public discussion, into claims about all ancient Egyptians. Cheddar Man's likely pigmentation category became, in the popular image, an exact reconstructed face. In each case, there is a gap between, most likely, given current models, and proven in every visual detail. Poor preservation creates gaps, and people tend to fill gaps with assumptions. Missing data can be mistaken for mystery, or even for conspiracy. Older studies can become outdated as methods improve, and that is not a failure of science. Reanalysis is the mechanism by which the field corrects itself. For anyone presenting this material, the lesson is to keep the genuine tension between evidence and its limits, because uncertainty can be dramatic without being exaggerated. The most dangerous phrase in ancient DNA is not unknown. It is case closed. There is one more reason these findings became explosive, and it is not technical at all. Ancient DNA does not enter an empty room. It enters a room already full of living identity debates. Cheddar Man became entangled with British identity and with the imagined appearance of early Europeans. The Egyptian mummy genomes landed in the middle of long-running and competing claims over the heritage of ancient Egypt. The recurring error is the same. Genetic ancestry affinity is read as if it were modern ethnic identity. Ancient populations rarely map cleanly onto modern nations or modern ethnic categories. The statistical phrase "closer to" does not mean the same as. Percentages become misleading when they are detached from sample size and geography, as the figure of 3 genome-wide samples from one Egyptian cemetery shows. A mitochondrial haplogroup, which traces only one maternal line, can be wrongly inflated into a statement about a whole population's origin, as the Haplogroup X debate shows. The Anzic one case demonstrates a better path in which researchers consulted descendant communities and treated the remains as ancestors, not merely as data. The Haplogroup X case shows how fringe groups can seize on a single unresolved detail. The Polynesian-Native American case shows the opposite. DNA validating a voyaging possibility that had long been dismissed. Some of this difficulty has historical roots. Colonial history disrupted indigenous records, displaced remains, and removed interpretive authority from descendant communities. Archaeological narratives were often shaped by outsiders. When context is lost in that way, genetics can appear to be the only available evidence, even where oral traditions carry real knowledge. The constructive response is precision in language. Ancestry, affinity, admixture, lineage, and population are useful words. Race is not a useful analytical term here. DNA can challenge myths, but simplified DNA can also create new ones. Ancient DNA does not just recover the dead, it unsettles the living. It helps, finally, to set the competing explanations side by side, because the controversies are not all the same kind of disagreement. Consider 3 broad categories of explanation. The mainstream scientific view treats human evolution as a branching network, with repeated admixture, deep population structure, and the regional extinction of lineages. The archaeological and historical view holds that some anomalies reflect real migrations, contacts, or cultural spreads that are simply not yet fully visible in the archaeological record. The fringe or speculative view argues for lost civilizations, transoceanic founding populations, or deliberately suppressed origins. These are not equally supported, and labeling them honestly is part of the work. Apply the 3 lenses to the cases. The Sima de los Huesos mitochondrial puzzle can be tested against incomplete lineage sorting, archaic admixture, or the later replacement of an earlier mitochondrial lineage. All 3 remain within mainstream population genetics. The West African ghost ancestry can be explained by archaic introgression, by deep structure within Homo sapiens, or by an unknown fossil population still to be found. The Tibetan EPAS1 signal can be read as adaptive introgression from Denisovans, as the influence of an unsampled Asian Archaic group, or as an older retained modern human variant. Lake Mungo can be explained by contamination, by the survival of an extinct lineage, or by a radically early settlement model. Accepted theories tend to survive because they predict multiple independent lines of evidence at once. Fringe theories often depend on a single anomaly while ignoring the genome-wide context. Haplogroup X is the clearest example, a real anomaly attached to a weak Solutrean conclusion. ANZIC1 is a clean falsification case, where genome-wide data defeated a claim built on the style of stone tools. Why does so much room for competing theories exist at all? Because vanished populations, erased lineages, and lost sites genuinely leave gaps. Rising sea levels since the last ice age drowned the coastlines that early migrants and seafarers would have used, removing exactly the evidence that would settle migration and contact debates. Absence of evidence can invite speculation, but it cannot, by itself, prove anything. Future ancient genomes will keep ranking these theories by how well they fit the data. Intellectual curiosity is valuable. False balance, treating a poorly supported idea as equal to a well-supported one, is not. And the mainstream account is not the dull option. It is often stranger than the fringe claim. The strongest theory is the one that explains both the anomaly and the surrounding evidence. The mystery is not that science has no answers. It is that every answer opens an older door. Step back, and a single pattern runs through all 10 cases. Human history is not a clean tree. It is a damaged, interbreeding, migrating, and politically charged archive. Sima de los Huesos showed that anatomy and maternal DNA can record different histories in the same skeleton. The West African ghost admixture showed that unknown human populations may survive only as statistics in living genomes, with no fossil at all. The Tibetan EPAS1 gene showed that archaic DNA can become a life-saving adaptation. Lake Mungo showed that contamination can briefly masquerade as a revolution in human origins. Cheddar Man showed that a probability can be turned into a political portrait. The Egyptian mummy genomes showed that a narrow sample from one cemetery can be stretched far beyond what it can support. Haplogroup X showed that a real genetic gap can be exploited by a weak interpretation. The Neanderthal genome showed that an extinct human group can be, quite literally, an ancestor. ANZIC1 showed that a single direct genome can settle decades of archaeological argument. The Polynesian-Native American signal showed that oceans connected people long before any European record noted it. The common thread is the power of the method. Ancient DNA allows researchers to test claims that were once trapped in the study of bone shape, artifact styles, and inherited myth. The causes of disappearance recur across the cases: climate that destroys DNA, simple decay over time, the extinction of lineages, migration that scatters populations, genetic bottlenecks that erase variation, and colonial disruption that severed communities from their own records. Entire populations can vanish physically while leaving a faint statistical trace, which means living people carry fragments of histories that no written archive ever recorded. The field is still young. More African genomes, more Pacific samples, broader regional coverage within Egypt, and better tools for predicting physical traits all lie ahead, and major reversals are still entirely possible. That youth is a reason for caution as much as excitement, and ancient DNA should not be used as identity propaganda or treated only as mystery entertainment. Human history is not a pure line of descent. It is a record of contact, survival, error, and replacement. The most controversial DNA findings are controversial because they reveal a past too tangled for the old maps.